Welcome to my talk. My name is Martin Estel. I'm a professor of computing science at Simon Fraser University in Burnaby, BC, Canada. In this talk, I'm going to introduce you to our work on machine learning for precision medicine. I will focus on the high level motivations, the problems, the approaches, and I refer you to the papers for further details. And I encourage you to email you with any further questions. Precision medicine has the vision to allow more accurate diagnosis and treatment of patients by taking into account their personal factors, taking into account the individual genomic profiles, the environmental conditions, and any lifestyle factors. And precision medicine promises to improve the quality of healthcare for the individual patients, and on the other hand, also promises to reduce the cost of healthcare for our society. Now, lifestyle factors could include exercise, nutrition, mainly, and other related factors. Environmental factors include socio-economical factors such as education, income, and so forth, but can also include the climate. Now, in precision oncology, the precision medicine for cancer patients, the genomic factors are especially important because cancer is known to be a disease of the genome. Now, precision medicine has been proposed a decade or maybe 2 decades ago, and certain success factors have been identified. This is a figure here from a recent survey paper by Denny and Collins. They identify a few success factors, and one of them, the one closest to home from our point of view of data mining and data science and machine learning, is here mentioned: big data and artificial intelligence, singled out as one of the success factors of precision medicine, and that's of course the focus of our research. This slide gives a snapshot of so-called multi-omics data, multiple types of genomics data. What we see here is on the x-axis the different positions on the genome, a total of 3 billion for the human genome. This is only a small part that we are visualizing here. And on the y-axis, we have different types of genomics or multi-omics data. We have point mutations, we have copy number variations, we have gene expression levels, protein expression levels, and so forth. I'm showing this slide to give you an impression of how raw multi-omics data looks like, and also to make it very clear that it's impossible to manually analyze these data, to manually determine patterns in this data, even within a single genome, let alone within hundreds of thousands or perhaps in the future millions of genomes. So that definitely calls for machine learning methods for automatically analyzing multi-omics data. And in particular, the overarching main goal here is always to find patterns and relate the genotype to the phenotype. So how can we use the genotype to predict the phenotype? Now, in our research, we typically don't work directly with raw multi-omics data. We use fairly heavily preprocessed data, and I'm referring to this kind of data, biomedical data. So this is biomedical data, raw data being transformed in matrix or table representation. So in this example here of biomedical data, rows correspond to patients, and we have 3 different types of columns or attributes. We have drugs, certain drugs, information about drugs that have been prescribed to that particular patient. We have information about adverse drug reactions observed for this patient. And finally, we have information about biomarkers, here in this case, we have biomarkers for each SNP. We have recorded the particular value on the different alleles at this particular position of the human genome. So what are the special machine learning challenges for multi-omics or biomedical data, in particular in the domain of precision medicine? Well, on one hand, data sets with clinical information are very small in the sense that they tend to have few rows in the order of hundreds, perhaps 1,000 if you're lucky, sometimes even below 100. And one of the approaches to deal with this challenge is transfer learning. So we try to transfer knowledge from larger data sets to smaller data sets. The second challenge is that biomedical data tends to have far more columns than rows. So we typically know a lot of information about a small number of patients or other entities of relevance. So a typical approach is representation learning. We learn lower dimensional representation of the high dimensional input data that capture the essence of the input data necessary to perform the machine learning task at hand. Now, deep neural networks have emerged in the last years as a method of choice for representation learning, and we are also making heavy use of deep neural networks in our research. The last machine learning challenge that is quite prominent for precision medicine is the need for causal patterns. So we work with scientists and physicians. They do not just want to learn models that are very predictive, that make accurate predictions. They want to learn models that are based not only on strong correlations, but on causal relationships between the input and output variables, because as scientists, they do want to understand the mechanisms of disease, for example. And so there's a need for two different tasks here. Causal discovery means learning which of the variables have a causal influence on a given outcome. In causal inference, you assume that you know the causal factors already, the treatments that are relevant, and you want to estimate the strength of the effect of a certain treatment, all from observational data only, without having the luxury of performing randomized controlled trials. Now, we are computer scientists, and we focus in our machine learning research on the general fundamental problems, which we try to solve in a principled and innovative manner, but we do take applications seriously. And in the field of precision medicine, our driving applications have been as follows: patient stratification. How can we group patients into strata, subgroups of patients that will respond similarly to treatments and correspond to similar subtypes of a disease? Second, driving application, drug response prediction. Typically, given the gene expression profile of a patient, we want to be able to predict the response to a particular drug. Last driving application has been the discovery of genetic causes of adverse drug reactions, again, from observational data only. So now in my talk, I'm going to focus on two main topics: transfer learning and causal models and causal inference. So let's start with transfer learning. The general idea is to transfer a machine learning model from a large source domain to a small target domain, or to a target domain that has no labels, unlabeled target domain data only. There are various scenarios that have been investigated in the literature. Two of the most prominent ones are domain adaptation, where we have a single labeled source domain and we have some unlabeled target domain data. Domain generalization is the scenario where we have multiple labeled source domains, but no target domain data at all. Now, a very important, groundbreaking result that forms the basis of much of the transfer learning research was this paper by Ben David back in 2010, which proposed a bound for the target classification error. So for any classifier in their paper called hypothesis, so the error of the hypothesis H on the target data set is bound by the sum of these three terms: the source classification error of the hypothesis, the difference in the distribution between the source and target domain, and the difference between the labeling functions of the two domains. And right, I mean, so basically this labeling function is basically the conditional probability of y given x. And in the domain adaptation research, we usually assume that this conditional probability is identical in the source and target domain. So this term would be reduced to 0, right? This is the marginal probability of the features P of x. And so these first two terms remain. So in transfer learning, in particular domain adaptation, the goal is to minimize the source classification error and the difference between the distributions of the two domains, the source and the target domain. Now I'm going to briefly introduce one of our works on transfer learning, the method called ATL, which is short for adversarial inductive transfer learning with input and output space adaptation. Our driving application here was drug response prediction. So given the gene expression profile, we want to predict the drug response. We want to transfer from so-called preclinical data sets, could be cell line data sets or data sets from mouse model, PDX data sets. We want to transfer from those as a source domain to patient data as the target domain. We do need to perform input space adaptation because the gene expression profiles between cell lines and patients, for example, are similar but significantly different. Now, we also have this additional challenge of having to perform output space adaptation because different measures are used for drug response. There's a continuous metric of the drug response for cell lines and a categorical metric of drug response in patients. Our method for input space adaptation is a common approach, adversarial learning, trying to learn a domain invariant representation. For output space adaptation, we perform multitask learning. Now, this figure presents an overview of our ITIL architecture. We have here on the left-hand side our source data. We have our target data. The first subnetwork of our neural network is a feature extractor, which here at this level produces a latent representation, a feature vector for the input. This feature vectors here, this is the same, these are the same nodes here. So these feature vectors are input for our multitask subnetwork. This multitask subnetwork has some shared level here and has some task-specific towers. One for the regression task of predicting the continuous drug response values, and for the classification task, predicting the categorical drug response. And yeah, so we have also the adversarial subnetworks that are in charge of the domain adaptation. So we have here a global discriminator which tries to predict whether a given feature vector comes from the source or from the target domain. And we also have class-wise discriminators which do this particular discrimination task not for all data as a whole, but specifically for one particular class. And the adversarial idea is that basically the feature extractor and the discriminators compete with each other. The feature extractor tries to learn representation that fools the discriminators, and the discriminators try to identify the source domain, the domain, whether source or target domain, from which the feature vector comes, as accurately as possible. So some more details: that multitask subnetwork employs extracted features for both the classification and the regression task. The classification network allows us to assign binary labels also to the source samples that have only continuous labels originally. Now these class labels are necessary for the class-wise discriminators, and the global and the class-wise discriminators try to distinguish the domains. And this results in the following loss function consisting of 4 terms: a regression loss on the source data set, a classification loss on the target data set, and then the losses of the global discriminators and the class-wise discriminators, all combined through some suitably tuned hyperparameters. I want to mention, at least briefly, some other work on transfer learning. In some cases, additional unlabeled source data is available, and we've investigated how to exploit such unlabeled source domain data to improve domain generalization. Our approach uses an objective function that combines a supervised loss for accurate prediction, an alignment loss for generalization, and the consistency loss. This is the additional loss for the exploiting the unlabeled data, and consistency loss for learning from unlabeled data. This was our Nature Machine Intelligence paper. We are also working on calibrated machine learning models. What is a calibrated machine learning model? Well, this is a model where the confidence of prediction agrees more or less with the accuracy of prediction. So, for example, if we have 100 predictions of a class with 90% confidence, then 90% of these in 90% of the cases, the prediction should actually be correct. So the accuracy of these 100 predictions should be 90% if all of these 100 predictions have a confidence of 90%. Now, calibration of machine learning models is crucial in mission-critical applications to build the trust, and in particular also in precision medicine. Now, there are existing methods for calibrating machine learning models, but they struggle with out-of-domain data, which we have to deal with in the context of transfer learning. And we are currently exploring methods for out-of-domain distribution calibration for the sake of domain generalization. Moving on to my second main topic, causal transfer. This is still one approach for transfer learning, causal transfer learning. So causal relationships are expected to transfer better between domains than relationships based on correlations. I mean, causal relationships are representing certain mechanisms. They're shared across different domains, but correlations could be spurious and may not transfer well between the domains. So that motivates the use of causal approaches to transfer learning. Now, the approach works as follows. We learn a structural causal model, basically a Bayesian network, directed acyclic graph with the corresponding parameters of the conditional probability distributions. So from that structural causal model, we extract the Markov blanket of the response variable. That is very useful because given the Markov blanket of a variable, the variable becomes conditionally independent from all other variables. So the Markov blanket in principle includes all of the information necessary for making accurate predictions. Now, in practice, the Markov blanket is hard to discover for high-dimensional data. which means that it may actually not be good enough in practice for making accurate predictions in either the source or the target domain. So the approach for causal transfer learning that we are currently exploring is to use all of the features, but to penalize those that are not part of the Markov blanket, so to prefer features in the Markov blanket, but to give other features a chance to contribute to the prediction. Okay, so now indeed, moving on to the second main topic of causal inference. The goal is the estimation of the effect of a known treatment or known causal factor T from observational data in the form covariates, which treatment was assigned to this unit, and the observed outcome for that given treatment. Now, causal inference from observational data is hard because of the presence of selection bias. And selection bias arises from the existence of confounders, variables that influence the treatment assignment and the outcome. So, for example, age could be a confounder that determines which drug a patient receives, and it's also going to influence the effectiveness of the drug. So, in the presence of confounders, the conditional probability of T given X is independent from the unconditional probability of T. That's different from randomized controlled trials where these probabilities agree, and that causes the problem. And that suggests and implies that a model trained on the factual outcomes on the observational data will be incorrect when predicting counterfactual outcomes. Why? Because the factual outcomes recorded in your database record the joint effect of the confounder and the treatment. So the approach, some of the approaches for causal inference are based on the so-called sufficiency of the propensity score. This is one of the methods here from Shi et al., so-called dragonnet. And this is here the formulation of the sufficiency of the propensity score, which states that if the treatment effect is identifiable, given the covariance X, then it is also identifiable given less information, given only the propensity score, the probability of treatment given X, and that motivates here the architecture of DragonNet. So we have here the covariance X as input. We have here one subnetwork for each of the two cases, no treatment or treatment. And so we learn here latent representation Z that can predict both the propensity score, the probability of treatment for this particular unit, and then also allows you to predict the outcome of treatment and the outcome of no treatment. Now we have investigated causal inference for multiple Boolean treatments, not just one Boolean treatment. We have developed a method for multi-treatment effect estimation. It makes the ignorability assumption that there are no unobserved confounders. That's a very common, somewhat limiting, but very common assumption that, for example, DragonNet also makes. So we assume that outside of the covariance X that we do observe, there are no other unobserved confounders. Now, our method is a so-called multi-gate mixture of experts. We have K plus 1 prediction tasks. We want to predict now K propensity scores and the outcome Y. We have experts, K plus 1 experts for these particular tasks. The experts are combined through a trainable gate function. Now, the strength of our approach M3E2 is that the network grows only linearly with a number of treatments, not it doesn't grow proportionally to the number of possible combinations of these different treatments. So it scales to a large number of different treatments. Now, the price that we pay is that we assume a linear combination of treatment effects, which is somewhat limiting but not uncommon assumption that overall treatment effect is a linear combination of the individual treatment effects. This slide provides an overview of the M3E2 architecture. We have here at the bottom a subnetwork that learns a latent representation. We have then here the different experts. We have here the gates, trainable gates that combine the result of the different experts. Now we have then here, I mean, this is an example for only two different treatments, k equals 2. So in this case, we learn two latent representations, H1 and H2, one for each of the treatments. We combine this in a linear manner to the overall latent representation of the input, and this capital H is then used to predict the outcome of a combination of treatments, and these latent representations here, H1 and H2, similar to what DragonNet does, allow you to predict not only the latent representation H, but also the probability of these different treatments. So this last layer is our M3E2 outcome model. Yeah, we are currently working on continuous treatment effect estimations and starting with a single continuous treatment that produces a new challenge, because now we have an infinite number of potential treatments, not just two alternative potential treatments. So we cannot adopt this common approach. We cannot have separate outcome prediction subnetworks for each of the treatments. We adopt an information theoretic approach for continuous treatment effect estimation based on the following intuition. We want to learn a latent representation Z such that the probability of treatment is independent from Z. That's similar to what we have, the property that we have for the original covariates in randomized controlled trials. And of course, we still want to be able to predict Y. So given, so the combination of Z and T together should be predictive of Y. Now, in our information theoretic approach, we use the mutual information. So on one hand, we want to minimize the mutual information between T and Z, and we want to maximize the mutual information between Z and Y given T. And this is all with respect to the encoder parameters. So we have a neural network with parameters pi that encodes the input X into the latent representation Z. Yeah, I do want to mention some other work on causal discovery. That's now not causal inference for known treatments or causal effects. This is work on causal discovery, discovering causal influences on some given outcome variable using observational data only. So this was some work that we have, a series of work that we've done in the last few years. Our approach was to use data mining to discover potential causes, hypotheses, then test those for causality by employing quasi-experimental design. The idea of a quasi-experimental design is you determine a subset of the observational data that in a certain sense corresponds to a randomized controlled trial. So for example, paired match design, where we match 5 pairs of examples, one being treated, one not being treated. And then last but not least, we need to correct for multiple hypothesis testing. We developed a series of methods. First method that allows you to discover single causes. Second method that looked into temporal databases. like databases from longitudinal studies. So here we developed a method that allows you to define temporal treatment patterns or temporal causal patterns from observational data. And the last work allows you to discover causes and corresponding strata based on the observation that the mechanisms of a certain that cause a certain outcome may be different in different subsets of the input data. In different strata of patients may correspond to different subtypes of the disease, and they may have different causal mechanisms. So here we simultaneously find the relevant subsets or strata of the data of the patients, for example, and the corresponding causes of the given outcome in this particular strata. Yeah, so in closing, I do want to summarize our ongoing and future work on precision medicine, more specific machine learning for precision medicine. So we continue to explore this topic of causal methods for transfer learning. We are actively investigating methods for model calibration for out-of-domain distribution data for transfer learning. We are investigating causal inference of continuous treatment effects. There's some follow-up work that we will be starting to investigate soon. We do want to develop causal inference methods with confidence intervals, methods that do not just produce a point estimate of the causal effect, but a certain confidence interval that will allow us to make the method more robust and more trustworthy. I do also want to mention briefly two lines of research that are not directly related to the other work that I've presented in this talk, but they also fall into this area of machine learning methods for precision medicine. These are works on the analysis of single-cell RNA-seq data. And in single-cell RNA-seq data, we have a matrix where rows correspond to cells and columns correspond to genes. And we have entries that represent the level of expression of a particular gene in a single cell. Now, this matrix tends to be very sparse. That's one of the grand challenges here in this domain. So we have worked on methods for cell type discovery. So we try to discover cell types within this sample of cells. And we annotate all of these cells with one of the known or novel, just discovered cell types here. So this would be the cell type column here. And then in the next step, we are using this single-cell RNA-seq data together with the cell type information for phenotype prediction. So now we have a sample of cells, for example, from a tumor sample. Now we want to predict the phenotype of that particular sample. So for example, is that sample, this tumor, going to develop metastasis or not? So we want to do phenotype prediction based on a sample of a single-cell RNA-seq data. So this is all, I think we have worked on this now for almost a year. We are about to publish papers on this topic. Yeah, so this concludes my talk. Here are my references. I thank you for your attention. I refer you to the papers for more details, and please email me if you have any further concrete questions. I'll be happy to discuss them with you. Again, thanks for your attention.